Thank you very much, Beth and Alexis, for the opportunity to share my latest research in progress with you guys here today. So today I'm going to talk about some metagenomic data from a tropical serpentinizing system. Thank you. So we've already heard about the process of serpentinization multiple times today, so I won't take too much time here to belabor the points. However, as far as astrobiology is concerned, it's essentially a process where we've got minerals and water coming together and producing life molecules, hydrogen, methane, and small organic carbons. And so from an astrobiological point of view, that has implications for both the origin of life here on Earth and potentially life on other planets. One of the best characterized sites of serpentinization is the Lost City hydrothermal field, which is located off of the Mid-Atlantic Ridge. And the communities there are dominated by methane cycling archaea on the interior of the chimneys, and they're called the Lost City methanosarcinales. And then the exterior of the vent chimneys are slightly more diverse and contain aerobic methanotrophic bacteria. And what this suggests is that methane cycling by microbes is incredibly important to communities in the Lost City. This is a map of some sites that our research group either has projects at or collaborations with other groups at. And we've been studying serpentinization in a continental setting for years. And so these results from the Lost City beg the question, is methane cycling by microbes important in these continental sites of serpentinization as well? So methanogens have been detected both at Oman and the Ligurian ophiolite in Italy using either 16S or metagenomic data sets. In Portugal, the tablelands ophiolite in Canada and the Coast Range ophiolite in California, they have not been detected. And that's either from 16S data sets, metagenomic data sets, or a combination of the two. And I just want to use this moment to plug a poster this evening. If you'd like to hear more about the tablelands ophiolite, please see poster 3311 by a technician in the Brazelton lab. And so for the remainder of the talk, I would like to talk about this one site in Costa Rica and whether or not we see methane cycling going on there. So the Santa Elena ophiolite is located in the North Pacific corner of Costa Rica. A paper by Sanchez Murillo et al. Looked at various geological and geochemical and microbiological parameters at a variety of sites at this location. Today I'll be talking about sites located right here. So two sites in particular. We've got Spring 9 and a very close proximity upstream site for a background sample. Spring 9 has the typical travertine calcium carbonate deposits that you'd expect when the high calcium fluids from serpentinization come into contact with the inorganic carbon in the air. And despite their very close proximity of a couple of meters from each other, they have different geochemistries. Spring 9 has a pH of 11.5 and an abundance of hydrogen and methane, whereas the background sample still has an elevated pH, but it's much lower and has less of these volatiles. And bioenergetic calculations have suggested that methanotrophy and methanogenesis are both favorable at both of these sites. So now to get into a little bit of the microbiological data. A lot of continental serpentinization sites have either no or very low abundance of archaeal taxa. And so here we looked at some quantitative PCR data, and I've highlighted here the two samples that we'll be talking about in this talk. And while the archaeal While archaea are less abundant than the bacteria, they still make up a substantial portion of the microbial communities, which is contrary to what we've seen in other sites. To explore the methane cycling a little bit further, I mined some 16S rRNA amplicon data sets to look for potential methane cyclers. Here we're looking at the bacterial communities. And I did not want to do that. I just want to orient you here that the units are the percent of the total bacteria community, but the axis goes from 0 to 10%, and that's because these were not in very high abundance at all. We see some aerobic methanotrophs in spring 9 and a slightly higher diversity and higher abundance in the background sample, but it still makes up less than 1% of the total community. The archaea tell a slightly different story. Again, we're looking at the percent of the total archaeal community, but here the axis ranges from 0 to 100%. In the warm colors, we're looking at methanogens, and in the cooler colors, they're putative methanotrophs. And so here we can see that in our background, in our high pH spring 9, the methane cycling archaea make up between 40 to 80% of the total archaeal community. And in the background sample, they're still significant, making up about 30 to 40% of that community. So since not everybody in the room right now is a molecular biologist, I thought I'd take just a few moments to explain some of the methodology for the data I'm about to present. So we use metagenomic sequencing, and we filter the natural microbial communities. We then extract DNA and chop that up into little pieces that can be sequenced on a high throughput sequencer. The ultimate goal for metagenomics is to try to reassemble complete genomes of individuals from the environment. And so we take these small little pieces of DNA and try to put them back together in larger chunks through assembly methods. Those larger chunks of DNA are called contigs. Binning is the step where you try to put back together these genomes, either complete or more likely near complete. And so here, this represents potential individual organisms where you can then annotate and predict their different metabolic pathways. And so this is an example of the assembly from spring 9, the high pH spring. From this assembly, we have 11 metagenomic bins. 10 of them were classified as bacteria. One was classified as unknown, which means that it wasn't even able to be determined at the domain level. And the coverage represents how complete, the completeness represents how complete these genomes are, since we're trying to obtain ideally full genomes. And so you can see here we have a wide range from 17 to 83% completeness, which at the lower end is not great. And so with these bacteria, I should point out that number one, none of these were identified as explicit methanotrophs, and also we don't see any archaeal bins here. However, as I mentioned before, from this metagenomic data, we can also not only look at who's there, but what their metabolic potential is. And so this is the methanogenesis pathway. These numbers here all represent potential genes in the pathway. The genes highlighted in blue were found in both the background and the high pH spring, whereas the genes highlighted in orange were found exclusively in the high pH sample. And I should note that there were no genes involved in methanogenesis that were found exclusively in the background sample. And so what we can see here is that we have complete methanogenesis pathway in the high pH spring. Also, there were no, I searched for bacterial methanotrophy genes in these metagenomes, and none were found. So as I mentioned, there's been a lot of research going on in the past few years into the microbial diversity and metabolism in continental serpentinization sites. And from that, some models have been developed. Some things that are found almost universally is an abundance of these beta proteobacteria, and also anaerobic Clostridia. And this data set was no different. There were an abundance of both of those groups. One question that still remains is what the role and abundance of these archaeal methanogens are in this system. We still don't have an answer to that, as they're found in some sites and not others, but we're beginning to dive into that now. So just to summarize, the evidence for methanogenesis at the St. Helena ophiolite include bioenergetics, archaeal 16S data, both from qPCR and amplicons, and full methanogenesis pathway in the spring 9 metagenome. However, no archaeal bins were found in that metagenome. And I just want to take a quick moment to explain that assembling metagenomes is much like taking hundreds of puzzles and spreading out all the pieces and trying to reassemble those puzzles again. And so, well, in this go-around, the puzzles didn't come back together very well. There are other methods to try to improve those bins. So my next steps are going to be to try to optimize the metagenomic assemblies and try to get more complete and representative bins. Additionally, we have metatranscriptomic data for both of these samples, and metatranscriptomes can tell you not only what genes are present in the environment, like the metagenomes, but they can tell you which ones are actively being expressed by the microbes. So stay tuned. With that, I would like to thank my co-authors and collaborators, my funding source of the Deep Carbon Observatory, as well as everyone who helped out with data analysis, sequencing, and sampling.
Thank you, Katrina. We have plenty of time for questions if folks want to come up to the microphone or stand up and speak really loudly.
Yes, that's very nice. So one of the persistent issues in genome analysis is unassigned open reading frames or open reading frames with unknown functions. Do you find in your various samples from different environments greater or lower abundances of the hard to assign stuff that might indicate that whatever they're doing, this environmental condition needs to make use of them?
Yeah, that's always a problem. So anytime that you're trying to assign, whether it be taxonomy or in this case function, you're limited by the databases that exist, and the databases are limited by what is already known. And so I think even in E. coli, about 40% of the genes are still hypothetical, meaning they're a gene, but we don't know what it does. And that number just goes up as you get into unexplored environments. Off the top of my head, I don't know what percentage of our genes are hypothetical, but it's definitely an issue. Cultured representatives can help us address that, but.
Is it fairly consistent across the different samples, sources, or phylotypes?
That's a great question that I don't know the answer to off the top of my head.
Katrina, I have a question. The fact that you see some of these archaea, I wonder if it's related to other parts of the chemistry of these waters in addition to just the methane and hydrogen. Particularly, what is the sulfate concentration like in these fluids?
Yeah, I know that some measurements were taken. I don't recall what they were off the top of my head, but we're beginning to look into some of these other electron acceptors at these sites to answer some of the questions.
Any other questions for Katrina? Oh, there we go. We got one.
Hey, Katrina, thanks. So do I understand it correctly that the methane cycling genes were found just in the random metagenome, but they weren't recovered in any bin?
No, actually, so the annotations were done on the bins, which raises a question of the validity of some of the bins, that those genes were all archaeal, but all of the bins were identified as bacteria. And so I think what we likely have is some archaeal contamination within those bins. So it's promising that we see the full pathway, but I think the bins need to be reassembled to try to get at that a little more.
Or you have a Nature paper.
Yeah, exactly.
All right. Well, thank you very much, Katrina.